So it is a difficult task, right? We can all agree, difficult multi-objective task, right? So, but now we have these powerful new tools. So we can maybe ask ourselves a little bit harder question, a more realistic question relevant to patients. So with the ensemble of conformations that are available to any given protein. But I love this introduction to the thought of complexity, because the other simplicity we have in looking at a target is we're looking at it, if not in vacuum, we're looking at it in buffer water. Okay, but in my view, cells are very crowded, very social environments, right? Proteins interacting with it, that's how the cell works. And with respect to complexity of conformations, and one system comes to mind is G protein coupled receptor getting to the surface, being signaled through, being taken up through different ways, right? Biased pathways. Where are we with maybe surfacing new insights around partial agonism or signaling through, beta-arrestin pathways versus one of the G protein coupled pathways? Are those kinds of insights coming? Okay, are they being delivered now through any of these methods, or do we know if anyone's looking for them? If we look for them, probably we will be able to find a way to achieve them.
For sure, it's changed a lot the number of possibilities that you have to interact with the target, as you said. So in the past, we have activate or inactivate on the orthosteric binding pocket. Then we found allosterism. That allosterism is the key for biasing, because you are doing something different. Then we see biasing within the orthosteric binding pocket. And now we can have, for example, was exploded, I think, the technology of covalent inhibition, because then you can target one single amino acid. Now we can target a plethora of amino acids. So it means that we can apply this as a technology almost wherever we want in a binding. We don't need anymore this cysteine waiting for us, because the chemistry evolved in a way that we can put this feature. So if you have problem of residence time, you can move to have a covalent reversible or not reversible inhibitor. So I think now we can design the kind of activation or better inhibition of the target in a more complex way. Also, as you mentioned, degraders, now we have protein-protein interfering, we have protein-protein interactors, chaperones. So there is a plethora of things now that we can do with, out of I block anything, or I activate anything, I modulate things. So I think that is really changing the approach, because now that you have so many ways to do, the design and the strategy that you put in place, what you want really to achieve. Because maybe when you stop anything that you rise side effect, then you have immunodepression, like happens in many drugs, are very good in many cases, but then once you have stopped anything related to the immune system, you enter in another kind of problem. This is the reason why we move from antibody to small molecule, because small molecule is a softened way to modulate respect an antibody. So there are many things that can be done today in the strategy. The important thing is to understand exactly what I want. So what I want to balance, what I want to achieve in terms of results.
Right, so that's interesting. It says that really with surfacing enough insights, it will help to tell us which therapeutic modality is really the best in class way to modulate these targets.
And as an example, when antibody comes out, biologics, now at that point they say biologic will remove small molecule from the market. And now what we're seeing that the second generation of antibodies are small molecule, because then can be orally variable, is not so aggressive. Now I can modulate. So I think when you put the complexity, in that case it's a switch on, switch off. Now with small molecule you can modulate more. So then you understand which is the right balance between. So you open an antibody and counterfactual, the evolution of antibody is again an old-fashioned small molecule, because then you can tweak the system much better.
And I think one thing that just coming to the conclusion as I've listened to you is, what is a target? A target is an ensemble of conformational states, many of which are addressable if we only think that we can do it and we have the tools to do so. A target is a modulator of a phenotype acting through potentially multiple mechanisms or multiple combinations of mechanisms, and bringing us to new intellectual vantage points, how to think about our craft, I think is really important. It enables people to do these things.
The complexity of biophysics, or mostly because it's not even biology, is the working cycle of a specific target. And this is where most of the models fail today to predict anything, is like some targets, a genetic mutation, one single amino acid can give you death or life. And you cannot predict that with models that can predict the structure or even the working cycle, because it's so minute. We are studying ion channels, for instance, and we have channelopathies, development channelopathies that are basically diseases related to some ion channels in which we have one genetic mutation in the working cycle, and it gives you epilepsy in kids. So it's really development disorders, and so now you have to have models that are physics informed. Why this single amino acid? And if you don't have the whole story about how is the working cycle of a channel that will conduct ions and be able to allow you neuron transmission, this is where AI fails today, because we don't have enough data and knowledge about how it is. And you have to put in your models physics-based strategies to be able to look at these little details at the level of an amino acid, sometimes of an atom or two. So it's interesting.
Yes, it's true. And I think it's so true for all the genetic disease. So you have a very single modification, but the effect, like in epilepsy, is huge. So the perturbation of the single modification of the amino acid produce a multifactorial chain of events. So when you have to restore, and in the case of genetic disorders, most of the time you cannot act on the protein because the protein do not fall. So nothing you can do with the real problem. So you know where is the smoking gun of the system, but you cannot act on it. No. And then you have to address the problem of a target that maybe in this case, in some cases, is really undruggable by the fact of the mutation. But then you have to act globally to restore the entire effect of the disequilibrium of ions in the cell. No. So there are some cases that are successful, like cystic fibrosis, then they were able to restore the effect of the single target. But in many cases, they have not the chance, again, with the mechanism of action. No? For cystic fibrosis, the golden molecule is a chaperone able to refold partially, and put the receptor where to do and gain some functionality. Now, this is completely unexpected at the time we are thinking activate or disactivate things. So still the complexity of the system and the strategy that you put in place. At the end, we know that for cystic fibrosis, now we arrive to 4 drugs. Now, to cover the entire set and to restore as much as possible the functionality of the system, not of the target. Now, so this is for me a great example how you can combine one by one, and then have a therapy that address the pathology starting from a single simple, not that simple at the end, event.
So it's the same for the modalities we see today, appearing new ones. Of course, antibodies, small molecule is one, and we have PROTACs, we have binders, we have people imagining new strategies, and it's really a big effort. Sometimes you don't need even an external partner or actor on that. In some of the projects we are working on, for example, in this channelopathies, if you know that some molecules are producing, for example, a gain of function, what you need is to reduce just the number of those translated. So you can use technologies such as antisense oligonucleotides, so the protein is not even produced at the level where it is, and it lowers the effect. So you get rid of the disease without even acting. You don't find a drug, you don't need a drug, you just make sure with a different technology that this molecule will not be at least translocated to the same ratio. So there is a hope in the and how smart are people in the field to uncover new ways. But we go back again to the same point as we said before in the beginning. It's like you need tools that increase your potential of gaining knowledge. That's AI assisting existing or more traditional ways. And then you need understanding of the effect, not just mimicking to say who does like what. So you have to have that physics-based modeling and knowledge, more knowledge acquired most of the time by combining technologies such as modeling or and experiments, of course, right? So
yeah, it's thinking like in terms of being able to apply physics-based approach, highly quantitative approaches. It's maybe like over the last 10 years where we don't actually have to do the hardcore quantum mechanics calculations anymore. We actually have DFT and other techniques which are computationally much less intensive, but yet very, very good, right? And at the same time, when we do have to do compute, our ability to compute has gone up by log units. Moore's law continues to kind of hold further. So that's really, all of that has happened in parallel. It really enables the kinds of things you've spoken to. At the same time, then we are learning to accept the complexity of biology. How do we extract from the accept as the complexity ways to modulate it and to bend with it and say this actually, if we think in this way, it's actually more like biology. And so with that latter point, I'm wondering, you end up with a complex system. And with these methods to, surface these kinds of profiles, right, for their really therapeutic hypotheses, are there techniques available now, or will they become available, where there comes an experimental design to validation along with it? Like, here is a roadmap of, experimentation that will give us experimental confidence that, this is the target, or these are the targets, this is the target site and the type of target modulation that actually will provide this effect.
On my knowledge, I think that the most advanced approach now is the project on virtual cells, like Isomorphic Labs is doing, like other company. So the point is to understand if you are already able to model in a certain way a cellular system that can support the design of the experiment, because we have a mechanicistic, so a mesoscale. No? So you start from the atomic simulation, and then you correlate with a mechanicistic model that control the effect. And there are activities now in companies to try to generate the data that enable the design of virtual cells that require a tremendous amount of data, because it's, as you said, very crowded system. There's no water with some protein within. No, it's a bunch of compressed object. But at the end, Isomorphic Lab said, no, is a matter of information sharing. So all these stuff do interact and say something to each other. So at least there will be a way to understand this interaction, this communication, and try to model. Let's see, it can be in 5 years, it can be in 15 years, we don't know. But I think that the direction is there. No, we have to see and change the scale and say, what is missing? Like when we decide to go to the moon. So we have not the technology, we are not able to do, but we have to go there. I think it's the same here. No, I don't know what we have to do, we don't have the data, but we know that we have to go there if we want to solve the problem of patients. No? And so the trajectory is there. And the consciousness of being there is moving the investment in that direction.